Hi everybody, I'm Dr. Munakal, Assistant Professor of Biopharmaceutics. We will continue today to talk about biopharmaceutical development and manufacturing of biosimilars part 2. We have learned from the previous lectures about the importance of knowing and understanding the reference drug and to choose the right expression system that will produce a protein that as close as possible to the reference drug. And we learn also about how to organize and to deal with the upstream and downstream processes and the scale up of the production for the biosimilar drug. Today we will continue to know more information also about development and manufacturing of the biosimilar. We will get started with the effect of raw materials on the product quality. Maybe it seems that it is overlooked, but still it's very important is assessing the critical raw material such as the culture media component, process buffering chemicals, excipient. Why? Because all of these they have an impact on the quality and hence the biosimilarity of the protein. For example, some specific culture media additives like N-acetyl uronic acid NaNA can block the production of N-G-N-acylic acid, which is the hydroxylated derivatives of NaNA, and this is not found in human and will provoke the immune system and they are immunogenic. In the same times, adding this additive to prevent the production of NGNA can alter post-transitional modification. Also, the sugars that are biologically derived, which means that can be contaminated and produce and have inside endotoxin. These sugars, such as glucose that use in culture media, or even sucrose or triols that use in a final formulation. When we say about endotoxin, we talk about undesirable impurities that we don't want to have it, and it can also vary from lot to lot. Therefore, this is again emphasize on the importance of performing assay to these lots. Why? To get rid the availability of these undesirable endotoxin. In the same times, you have to have a careful consideration of the critical raw material should be continued throughout the all formulation development. So you need to keep checking the critical raw material and make the appropriate assessing assay for it. So um, uh, other things that we should take in our consideration, we talk about the sugar and how it can affect and has a potential to produce endotoxin. Also about the adding additives which can also affect the raw material. Also the oxygen. It's well known that oxygen produces peroxides in a solution of polyether, such as polysorbate twin. In the same times, as a pharmacist, all of us know that peroxides are highly reactive. And when we mean that it's highly reactive, it can abduct hydrogen atoms from the surrounding protein and cause protein degradation. So, how to contain the peroxides, how to have a very low level of peroxide and control it. Can be achieved by combining a source high, ultra high purity polysorbate meeting tight specification for lower level of peroxide drawing only from previously unopened container, in the same times also using a fresh solution. So all these together would be helpful in minimizing or maintaining a very low level of peroxide. Also, regarding effect of the raw materials on the product quality, like for any biopharmaceutics, looking or having animal-derived raw materials is not a preferable things and should be avoided. Why? Because the animal-derived raw materials have the high potential for the contamination. So if we couldn't avoid using animal derived raw, and there is no choices for that, and we have to choose it and use it. So there will be a risk assessment has to be done in order to make sure that the potential for this contamination um, doesn't have source of transmissible spongiform encephalopathies TSE, because we don't want to have this bad situation, because TSE is they are a group of progressive invariably fatal conditions that associated with the prions that can cause encephalopathies, a disease for the brain and the nervous system. So we don't want this to have it with the biosimilar drug. So a precise risk assessment should be done. So also there is other impurities we could have it in the raw materials, such as the one that we can get it from the transition metal ions that can act as a catalyst for the oxidation of sensitive amino acids such as methionine. Um, Moreover, there is a trend of using a single-use technologies. So these single-use technologies impose the utilizing of the plastic. Plastics, it is a new potential source of impurities. Why? Because as we know, the plasticizers that found the plastic, they could be leachables and extractables, and this is it's cause impurities and many problems. So these leachables and extractables can come from the plastic material. So plastic bags, they are widely used throughout the entire production process of biosimilar drugs, such as bags used to contain the cells and media of a production to others for storage of the buffers and other intermediates, bulk drug substance. So this is so important when we use plastic bags, to keep in our mind that there is a must of rigorously testing the plastic material for these leachables and extractables, that they are undesirable, and their effect on the biosimilarity of the drugs should be evaluated all the time. So, in order to optimize everything about the raw material use and their quality, the United States Pharmacopoeia has developed a rigorous series of tests um, for such materials for medical use. They call type 7 classification, and they are biologically compatible and suitable for use in patient. So in order to get to get rid of these leachables and extractables, the manufacturers, they are interested of single use system. They have to have the source. They have to have the source of plastic bags with a high purity. So also in the same time, the customers have the right to ask the suppliers of these plastic bags to provide them with all the data about the extractable and leachable that can be derived from the manufacturer, and they should also insist to know even the fine details that if these bags are produced according to the USB type 5 material. So um, as we'll continue to talk about the raw material, so testing and control of these residual process impurities such as hostile protein, hostile DNA, and protein chromatographic ligands such as protein A is the same for biosimilars as for novel biopharmaceutics pharmaceutical. So all of these things also should be tested and should be controlled. So um, as also we mentioned before, and we will reiterate about it, that the levels of such these impurities and residuals, or not about residual, should be comparable to that or lower than that of the reference drug. So again and again, it should the biosimilar it should be as close as possible to reference drugs in all aspects. So this represent a potential safety concern since biosimilar developers is relying on the positive safety history of the reference product. So if we deviate from this history, we will put the patient at risk, and we don't want that. We want to get all the details about the biosimilar drug to be close as possible as that for the reference product. So, after studying the effect of raw material on the biosimilarity, now we'll study about the effect of manufacturing conditions on the product quality and the control strategy for biosimilars. So, a tight control for the critical quality attributes is required through all the process of the development of upstream and downstream to ensure that the selected conditions for producing the biosimilar don't induce any structural changes that could result that the target protein of interest it no more of interest, it will be outside the range of the reference product. So many analytical methods should be available and should be done in order to maintaining the biosimilarity in all the steps in the production of the biosimilar drugs. So there is a specification need to be set, which they are according to the International Conference of Harmonization. Like for any biologic, biosimilars need to have these specification, which are they are setting specification for biological drug substance and the product. They are testing procedures and testing the acceptance criteria for biotechnological or biological product. This guidance document provide guidance on general principle for the setting and the justification to the extent possible of a uniform set of international specification for biotechnological and biological products to support a new marketing application. And if you want and interested to know more information about this specification, I already insert a link here in this slide, and to read all about this specification. But of course, this is not included in the exam, it's just for your information. So part of the parameters that are being tested, and also their acceptance would be reflected into the specific process that used to produce the manufacturing biosimilars, and they are not or they are differs from the originator. It is studying or testing the parameters like residuals from materials used in upstream media in the culturing, residuals from material used in downstream purification step, host cell protein, residual host cell DNA. Um, also there is many analytical comparability studies. These serve as a guidance why to establish a specification needed for the critical quality attribute. This is in turn can help us in what? Can help us in making decisions and to the acceptable ranges in the acceptable acceptance criteria that's it for the critical quality attribute to ensure biosimilarity. So again, as we said before, that the biosimilars they have many extensive analytical studies. All they are around and they are about just to ensure biosimilarity. So these specification could vary depending on the type of molecule, whether it is recombinant protein or monoclonal antibody. So, if we want to talk about these differences, for example, for a glycosylated molecule, understanding the impact of glycan compositions on the potency, it is so important. Why? Because this variability is expected from lot to lot. So, glycosylation affect the potency of the protein. Also, the variation in the level of silylation can influence pharmacokinetic profile of the molecule. Also, for monoclonal antibody, for example, oligosaccharide, like core fucosylation, which is another important modification in glycan core structure. This is have an impact has an impact on FC conformation, which means that this will lead to lowering the binding affinity of molecule to FC receptors. So again, all of these specifications are needed to ensure the biosimilarity at all the production steps. Also, it is important to mention that these structural changes or features, um, we want these features to be maintained within the acceptable range. We couldn't like exactly the same, like, but there is like a range, there is acceptable range. We should maintain this feature within this range. How we can obtain that? By keep and continuous monitoring and ensure a sufficient controlled processes. Also, the amino acid modification of monoclonal antibody need to be controlled, as these modification also have an impact on the product potency. Such modification could be happen like methionine oxidation, asparagine deamidation. Also, an important aspect of setting this specification is the evaluation of stability behavior for biosimilar candidate compared to that of reference material. Stability behavior here we focus about the degradation profiles. This degradation profile can be obtained by side and forced degradation studies. Why it's important to do this type of these studies? That because it helps us to uncover the behavioral differences between the biosimilar candidate and the reference material. So, example of these behavioral differences, like the level of oxidation that could happen to the biosimilar product due to the metal catalysis from process residuals, and these will need additional step of control, because we don't want these behavioral differences to be exist between the biosimilar and the biological drugs. Also, here in this slide, also it shows an example of typical pores degradation conditions, like exposures to high temperature, light, low and high pH, and oxidizing conditions. These typical pores degradation could provide us with such information about if there is a behavioral differences between the biological drugs and the or the biosimilar drugs and the reference drug. So their impact on the protein, these behavioral differences and their impact on the protein should be evaluated uh, for both the biosimilar candidate and the reference material. How? In terms of accumulation of low and high molecular weight species, here we can use the size exclusion chromatography. We can also use reducing and non-reducing capillary electrophoresis sodium sulfate. Also, we can check for the changes in the charge species, and this is size species. This is we can use the cation exchange HPLC. Also, we can check for any conformational changes, which we here, and we can use circular dichroism, and we can check also for the potency and protein concentration using the UV. So there is a list of specification for a biosimilar BDS is presented in table 1, as we will explain it with a few seconds. So this table show generic specification. Like the first column is for the parameter that we want to check it. Here for the quality attribute that we are looking for, and here for the method. For example, for the identity, here, what's the quality attribute here? We want to check the primary sequence of amino acid. Here is the best used method is peptide mapping by reverse phase HPLC UV. For the content, we want to know content is reflected in the protein concentration. So we use UV spectroscopy at 280 nanometer. Purity, if we are looking about size variant, we use size exclusion HPLC. We can use capillary electrophoresis. If we are looking for the charge variant, we use capillary isoelectric focusing or hydrophobic interaction chromatography. If we are looking about the purity, about the level of glycosylation, we can use oligosaccharide mapping, normal phase HPLC with a fluorescence detection. We can use sialic acids by reverse phase HPLC with a fluorescence detection. If we are looking for potency, here we looking for the binding affinity. So specific assays should be done for the a process of related impurities, like we are here the quality attribute should be tested, residual process impurities that are and the method used specific to the process, like residual protein A. If we are looking to residual host cell protein, here also specific to expression system, we can use Western blot overlaid with antibody for a specific cell line. Also, if we looking for residual host cell DNA, here we use quantitative PCR to quantify the DNA level there. So the functional assay should be developed in a way very early. Why? To measure the biological activity from the beginning, to ensure that the biosimilar candidates are effective and suited for moving along the development process. So, as the functional assay used for testing monoclonal antibodies, one or more of these assays later it will become an integral part of this specification after appropriate validation. Why? To ensure assay performance. So, types of biological activity assays that are used for typical antibody therapeutics, it's like binding assays, ligand binding assays, competitive ligand binding, and also bioassays. Example of it: in vivo bioassay, in vitro tissue-based bioassay, in vitro saline-based bioassay, and reporter gene bioassay. So, um, as the number of lots manufactured using the same process increases, that means the result of all these parameters that have these specification in a process and the stability result, all of these together will create a data base in support in a process of validation, and this is will enable us to set a robust specification ranges for commercial lots for the biosimilar drugs. And finally, and most importantly, stay at home and stay safe. And how to stay safe from coronavirus? Remember always to wash your hands regularly, sneezing cough in a tissue, or sneezing cough inside your elbow in case you don't have a tissue. Also, at the end, bin it through your tissue away immediately. And please stay positive and keep your energy going on and never give up. And thank you so much. This is the lecture number 11.